Hi, my name is Deepak Sarkar. I'm a distinguished professor in the Department of Animal Sciences at Rutgers University in New Jersey. Today, I want to explain to you how alcohol affects epigenetic system to alter body functions, especially when we drink a large amount of alcohol. Scientists have clearly shown now the large amount of alcohol drinking affects various parts of the body, like brain, heart, liver, pancreas, and immune system, and causes varieties of diseases like cardiovascular problem, pulmonary diseases like pneumonia, bronchitis, cancer, fetal alcohol syndromes, and many others. This happened because alcohol affects our epigenome and epigenetic machineries. And that's what will be the focus of my talk today. What are the epigenetic machineries? And how do these systems affect our body function? Our body has about 200 cells and all contain the same DNA. How can they look and behave so differently? That is happened because of the epigenetic phenomena. What is epigenetic phenomena? It basically turns on gene or turns genes off. But how do they turning on and off make cells looking different, functioning different? Because each cell, specific group of cells, has a epigenetic code, which is just like a barcode. And when the epigenetic machinery read this code, makes the cell look different or behave different. Why do they do so? And how do they do so? And that happens because of the epigenetic phenomena or epigenetic alteration. So epigenetic phenomena, which causes genes turn on or genes turn off. And by doing so, it makes the cells look different or behave differently. The coding is just like a barcode, which tells when the epigenetic process goes through, which tells the cells to function one way versus other way, or cells look one type versus other type, like neuron versus cardiomyocyte or monocytes. So how does it happen, and what is these epigenetic phenomena or epigenetic machineries, and how do they work? So the epigenetic marks are two kinds of marks. One is a chemical mark, another is a protein mark. Chemical mark is basically a methyl mark, or is an addition of a methyl group within the nucleotides of DNA. As you all know, that we have 4 nucleotides, A, T, G, C. Within the cytosine area of the DNA, methyl groups get added, and that's caused methylation. In certain cases, the methyl group is also added in adenine nucleotides, but primarily it happens within the cytosine nucleotide. And cytosine nucleotides are often gathered with the guanine or within the GC island, or it also called as a CPG island. CPG island are within the promoter area of the DNA or the gene, as well as the gene body area of the DNA. And by methylating this area, it affects gene expression. Protein mark, on the other hand, is the changes takes place within this group of basic protein called histone proteins, which are wrapped around the DNA molecules and form nucleotides, and then condense and form chromatin within the nucleus. So epigenetic modification then happen primarily on these DNA within the histone, and so they call DNA methylation and histone modification. It also happens guided by the non-coding RNA or microRNA, but most of the research has been done with the histone modification and DNA methylation within the alcohol field. So my focus will be primarily on DNA methylation and histone modification. So DNA methylation, as I already mentioned, is addition of a methyl group within cytosines. And this happens primarily by enzymes called DNA methyltransferases. There are three kinds of DNA methyltransferases: DNMT1, DNMT3A, and DNMT3B. And DNA methylation or removal of the methyl group, it takes place by a protein called TET proteins, which are TET1, TET2, or TET3. So the DNA methylation, the way it controls the gene transcription by preventing the interaction with the transcription factor on the DNA or the binding of the histone protein on the DNA. So DNA methylation turns off or low transcription, gene transcription, and DNA demethylation will cause high transcription or turning on the gene transcription. Histone modification or chromatin modification takes place within the histone protein. Histone proteins are these basic protein, H2A, H2B, H3, and H4. They are wrapped with 144 base pair of DNA to keep as a chromatin molecule or nucleosomes. And the modification is primarily takes place within the N-terminal portion of these histone proteins. And this modification happen as acetylation of these histone protein, methylation, phosphorylation, or ubiquitination. Acetylation of histone protein usually turns the gene on. Methylation of a histone usually turns the gene off. However, methylation of H4 and H3, depending on the site of the methylation, some methylation of H4 turns the gene on, whereas most of the site of the H3 turns the gene off. Phosphorylation, on the other hand, function as the amino acid specific, turns on and off, and ubiquitination cause increased transcription or gene turns on. So why alcohol affects epigenome? Alcohol affect one carbon metabolites and produce this molecule called adenosine methionine or SAM. SAM is one of the source of methyl group for DNA methylation, as well as some of the histone protein methylation. Alcohol also get metabolized by acetaldehyde and form acetyl-CoA. Acetyl-CoA is the source for acetylation or acetyl group and help chromatin acetylation, which help for genes turning on. Methylation, on the other hand, causes gene turns off. So there are now quite a bit of substantial work in human as well as in rodent has been done to study the alcohol effect on our body functions. Primarily, the work has been done with the adult population as well as adolescents in fetal alcohol exposed subjects. And we are also conducting some of this study, primarily dealing with the DNA methylation. And I would like to present some of those data to show how alcohol exposures causes epigenetic modification to affect body function. The figures shown here is showing drinking alcohol may change the DNA methylation in adult alcoholic that leads to an increased craving for alcohol. This work has been done with Dr. Rajita Sinha at Yale University. And what we found here, we found that a gene called pro-opiomelanocortin or POMC gene, their methylation changes are positively associated with the amount of alcohol drinking as well as alcohol craving. So what it shows that more POMC gene methylation happens, more drinking takes place, more craving takes place. So there's a good correlation association between this POMC gene methylation with the amount of drinking and amount of craving. So that suggests that the epigenetic machinery can participate, can influence alcohol drinking and alcohol craving behavior in humans. We also studied this alcohol effect in fetal alcohol models in rodent. In fetal alcohol model in rodent, we use very similar to human second trimester of pregnancy and gave these pregnant dams alcohol, which give rise to 120 milligram per deciliter of blood alcohol concentration. When you do so, we also find that the fetal alcohol exposures in the dam leads to the altered POMC gene expression in the offspring, both male and female. There we find, just like the in human, the alcohol exposure causes increased DNA methylation and suppression of POMC gene, both male and female offspring. Interesting, however, very unexpectedly, that we find that this fetal alcohol effect on this POMC gene expression as well as POMC gene methylation bypasses this epigenetic machinery, which is very critical during the embryogenesis, where the most of the genetic modification and all those goes through reprogramming to correct those genetic alterations. But this effect, fetal alcohol effect on POMC gene, bypasses that and transmit to second and third generation. Those are the we studied, but it could bypass it longer. So this is interesting, and this is very surprising, because what it suggests that fetal alcohol effect not only goes through the first generation offspring, but it could passes on second and third generation offspring, grandchildren and great-great-grandchildren. So there are several other studies we conducted, and we found that the fetal alcohol effect on this multiple generation, not only with this POMC gene expression and the stress axis, but also on immune function, as well as immune-related diseases like cancer, anxiety, behavior, as well as the metabolic function. So the fetal alcohol effect, what we saw in animal, also been tested in human patients, human subjects. This work has been done in collaboration with CFAS, a consortium of fetal alcohol syndrome researcher, where we have obtained samples from the mother who has drank alcohol and gave birth to fetal alcohol exposed offspring or fetal alcohol syndrome babies. And what you find, just like in animal model, in mother who drank alcohol during pregnancy and gave birth to these fetal alcohol syndrome babies or fetal alcohol exposed babies, have a higher level of POMC gene methylation. This not only happened in mother, but also we found in children who were either fetal alcohol exposed or have these fetal alcohol syndrome features. This we found they also have the POMC gene increased gene methylation done by varieties of methods. First method is called MSP or methyl specific PCR. The other is called pyrosequencing. Both methods showed indeed these the fetal alcohol exposed children or FASD feature children have this hypermethylation of POMC genes. When we looked at their stress level by measuring a cortisol level, we also find the fetal alcohol exposed children have a high elevated level of stress. So suggesting, just like in animal model, suggesting that fetal alcohol exposures does indeed alter the endocrine phenotype like hyperstress response, as well as the epigenetic phenotype like hyper POMC gene methylation. One of the things we also studied to see whether or not changing the epigenetic modification by nutrient could affect this abnormal DNA methylation in these fetal alcohol exposed children. And the way we did this by feeding these fetal alcohol children with choline. Choline is a nutrient which in animal study has shown choline can alter DNA methylation process. And when we gave this choline supplement to these children for several months, what we do find that longer we give choline, more suppression of POMC methylation takes place and increase of POMC gene expression takes place, showing that choline might be useful to modify some of those epigenetic modification to reverse the alcohol effect. However, this study needs to be conducted further and has to be done not only in these POMC gene methylation, other methylations, as well as the behavioral phenotype of epigenetic modification. I have shown to you today that the epigenetic machinery is a critical part of alcohol effect on our body system. And learning about them really helps us to learn how alcohol can alter epigenetic machinery and impact the bodily function. And also, it gives us the hope that there may be new therapy we can develop from this epigenetic modification to cure some of these problems associated with large alcohol drinking. I appreciate you for listening to my talk. And finally, I wanted to acknowledge the funding from NIT.